Hello and welcome to CoolSchool Genetics की online पाठशाला. In this video, we will be discussing about Mendel's Law, the basis of your entire understanding about genetics. So let's get started with this video lecture. The first thing to know about genetics. What is genetics? So, genetics is a branch of biology concerned with the study of genes, genetic variations, and heredity in organisms. A nice, sophisticated definition for examinations. But what genetics actually is? So, talking about what genetics is, let us first understand what genes are. Let us start from the scratch, absolutely scratch. So, genes are the discrete units which are responsible for controlling any character in an organism. There are always two genes which will be a pair of genes, I can say, which will be responsible for controlling one character. Okay, so and those two genes individually would be alternative forms of each other, which can be known as alleles. What I mean by this is, for suppose there is a pair of gene controlling the height of the plant, and the tallness of the plant is represented by capital T, and the dwarfness or the shortness of the plant is determined by the gene small t. So capital T and small t are the alternative forms of each other, or the alleles of each other. Then, talking about variation. Genetics is all about variations. By variations, we mean the degree of changes, that is, the degree of difference between the progeny and the parent. By progeny, we mean the produced generation, the babies, I can say in a layman language. That is, you must be different from your parents as well. This is variation. Okay, so if we calculate it on the basis of certain degrees, it would be known as variation. Okay, and the characters you have got from your parents, that is inheritance, inherited from your parents. Okay, next is locus. So what do we understand by locus? Genes are present on chromosomes. So the location of the gene on chromosome is known as locus. Now there are two types of genes. Not genes. There are two types of alleles: the dominant allele and the recessive allele. What do we understand by dominant allele? The allele which will represent itself in every condition. That is, be it homozygous condition or heterozygous condition. Homozygous condition means that when the gene pair is similar, capital T, the gene pair is similar, is known as homozygous condition. And heterozygous condition means capital T and small t, when the gene pair is different. This is known as heterozygous condition. So a dominant allele or a dominant trait will be represented in both the conditions, be it heterozygous condition or homozygous condition. But a recessive allele will only be represented or expressed in homozygous condition. That is small t or capital T. When the gene pair is same, only then the recessive trait or the recessive allele will be expressed. I guess I am quite clear. Moving forward, next we have Mendel's experimental design. For the first time, statistical analysis and mathematical knowledge was applied to problems in biology. So, we all know that Mendel came up with certain ratios, phenotypic ratios, genotypic ratios. These ratios are a part of maths. So, it was for the first time that someone brought mathematics into biology. So, this is what we mean by statistical analysis. So, Mendel was the first person to combine mathematics with biology. Now, the other great question, or the other major question which the students ask, that is, why did Mendel choose pea plant? There are so many plants available, but why only pea plant? So uh, let me tell you, there are very many varieties of pea plant as well. So Mendel particularly chose garden pea, by scientifically known as Pisum sativum. Okay, so why did Mendel choose garden pea? Okay, so the reason is that pea plant is easy to grow, it bears flowers and fruits in the same year a seed is planted and produces large number of seeds. And also that a pea plant has many contrasting characters. You can see by the image we have used here that there are very many contrasting characters present in a pea plant. This is also a very big reason behind choosing garden pea. And the other reason is self fertilization takes place in pea plant, so it is possible to get a pure line of trait. What do I mean by pure line of trait? Pure line means that the character remains unchanged from parent to progeny, that is, no variation takes place. If the parent had round green colored seed shape, then the progeny will also have round green colored seed shape. So this is known as pure line trait. Okay, so acha, one more important thing. Mendel had no knowledge of meiosis and mitosis, so he did not know that genes segregate according to the chromosomal behavior. I won't explain this statement right now. Just wait for it, and by the time we end Mendel's law, I believe that you will yourself understand what do I mean by this statement. Till then, just remember this statement. I'll repeat it once again. Mendel had no knowledge of meiosis and mitosis, so he did not know that genes segregate according to chromosome behavior. Okay, so before moving to the next slide, I want to discuss all the contrasting traits of the pea plant. Okay, so the first one, okay, remember, shape is the character, and round, wrinkled are the traits of this shape. Oh, sorry, are the traits of this character. Okay, every character has two traits. Okay, so we'll go with this one. The first one is seed shape, which is round. Round one is dominant, and wrinkled, which is recessive. By dominant, I mean that it will express itself in both homozygous and heterozygous condition, whereas wrinkled will only express itself in homozygous condition because it is a recessive trait. Okay, so whenever I'm saying that it is a homozygous trait, oh sorry, it is a dominant trait, it is a recessive trait, I am talking genotypically, that is, I'm talking about the genetic constitution of the organism. Okay, next up we have seed color. The dominant color is yellow and recessive is green. Pod shape, dominant is inflated, recessive is constricted. Pod color, dominant is green, recessive is yellow. Okay, don't get confused between seed color and pod color. In seed color, the dominant color is yellow, whereas in pod color, the dominant one is green. Students most of the time get confused in this, so I am pointing it out specifically that don't get confused in seed color and pod color. The dominant color for seed is yellow and for pod it is green. Okay, and the next character is flower color, which has two traits. Purple is the dominant trait and white is the recessive trait. The flower location, the dominant one is axial and recessive one is terminal. And for plant size, the dominant one is tall and for recessive it's dwarf. Moving forward to the next topic, we have Mendel performed cross pollination in pea plants. So I told you some time back only that pea plants undergo self fertilization. So why did Mendel perform cross pollination in pea plants? See, Mendel was working on a hypothesis. He had a hypothesis that about genetics. Okay, so there are very many steps to test a hypothesis. So of course he had to, experiment with his hypothesis, and for that he needed variation in pea plants, because if we go for self fertilization will always lead to pure line. But to prove a hypothesis, we need to divert ourselves from the pure line as well. So Mendel performed cross pollination. What did he do is that he took out the pollens, he removed the pollens or the male organs from the plant before the female or the pistil part matured in that. How did he do that? He did it by the process known as emasculation, removal of pollen grains or the cutting of pollen grains from the plant before the pistil or the female organ matures is known as emasculation. Then he fertilized the pistil then he took the fertilized pistil of some other plant and artificially, or I can say in laboratory, he fused them, and then all the offsprings which were received, that were cross pollinated offsprings. Okay, so moving forward, next we have inheritance of one gene. Before we start inheritance of one gene, you need to understand about monohybrid cross. What do we mean by monohybrid cross? Mono, let's break the word to understand it better. Okay, mono means one, hybrid means the crossing, and cross means again mating. One and the same thing. So, when we take only one character in consideration to perform a cross or to mate, then it is known as monohybrid cross. Likewise, here we have taken only one character. Can anyone tell me which character have we taken? You can see that capital Y is the dominant trait. We always represent the dominant trait with capital letters and recessive trait with the small letters. So here from the picture it is very clear that yellow is the dominant trait. So can you tell me that is it the pod color or the seed color we have taken? So I believe that you must have guessed it till now, that it is the seed color we have taken. So we have taken only one character in consideration, that is seed color, for which the dominant trait is yellow color and recessive is green color. So after taking out the gametes, we have capital Y and small y. Generally there are two gametes, but because from this parent both the gametes would be same, so they have written only one gamete, and similarly here also they have written only one gamete. So when we cross them, we get capital Y and small y. So as I said, it is a heterozygous condition, and only dominant trait is able to express itself in heterozygous condition. So ultimately the seed color would be yellow. So this is our F1 generation, first filial progeny or the first hybrid generation. Okay, so just remember the term filial progeny. F1 is our first filial progeny. There is a important point to remember about F1 generation. F1 progeny generally resembles either one of the parents, and the trait of another parent is not visible. Likewise, you can see here also that the trait of only dominant parent is visible in F1 plant, that is, the seed color is yellow as here as well, whereas the green color has been hidden. This green color would again be visible in F2 generation. You can see here we have got one progeny in green color. So F1 parent resembles only one of its parent and masks the trait of another parent, whereas the hidden trait is once again visible in the F2 generation. Okay, so here you can see the genotypic ratio and the phenotypic ratio. By genotypic ratio we mean the genetic constitution, like the by genetic constitution whichever would similar things like capital Y, capital Y. So we have only one capital Y. So only one of the progenies having capital Y, capital Y. So it would contribute to only one. Okay, and likewise, the entire genotype ratio for monohybrid cross will be 1 is to 2 is to 1, whereas phenotype ratio is the observable characters of the plant of an organism is known as its phenotypic ratio, which is 3 is to 1. We have 3 yellow offsprings and 1 green offsprings. For all the monohybrid cross, genotype ratio would be 1 is to 2 is to 1 and phenotype ratio would be 3 is to 1. Moving forward, next we have the reciprocal cross, back cross, and the test cross. So, talking about reciprocal cross, as it must be very clearly visible by the name itself, reciprocal cross. This means to turn things upside down or to interchange. Likewise, just look at here. Parent 1 is female here and parent 2 is male in the first case. After performing reciprocal cross, parent 1 becomes male and parent 2 becomes female. That is, the genders of both the parents have been interchanged. This is known as reciprocal cross. Talking about back cross. So, what is back cross? When the cross is performed amongst F1 generation and one of the parent, irrespective of the fact that it is a dominant parent or the recessive parent, but any one parent, then it is known as a back cross. Okay, you can see here that our F1 generation is capital T small t, that is a heterozygous tall plant. When it has been crossed with its dominant parent, that is capital T capital T. So this is known as back cross. Now why do we perform back cross? So we perform back cross to recover useful characters from the parental population. What do I mean by recover useful characters? They would be visible in F2 generation, right? But there are certain characters which we want they should be visible in each of the progeny. We want to make them pure line, I can say. So what we do is that we make them cross with their parent, so that once again we get a new progeny in which this particular trait or any particular trait becomes stabilized. Now here you can see that capital T, that is the tall height, has been stabilized in this progeny, right? So this is what I meant by back cross. If I would have crossed F1 generation only, like by the normal norms, then I would have got one small t as well in the F2 generation. But in this case, I will not get any small t for the F2 generation, right? So that is what the use of back cross. Moving forward, next we have a test cross. So test cross is generally used to find the genotype of an unknown organism. Okay, of an unknown organism with a known phenotype. We all know that purple color is a dominant trait. Okay, but when you see a purple flower, you cannot be sure that it is a homozygous purple or a heterozygous purple. So what we do, we cross it with a recessive genotype, that is a white color flower. Okay, and how we do it? We assume two situations. One, that the unknown purple flower has a genotype of homozygous dominant purple flower, that is capital P, and the other situation in which the genotype is heterozygous dominant, that is capital P and small p. In this case, in case 1, all the progenies will have purple flowers, whereas in case 2, half of the progenies will have purple whereas half will have white. So by looking at the entire set of progeny, we can find the genotype of this unknown flower also. We never talk about one single flower. This is so obvious that a pea plant will not just give one flower at a time, it gives many flowers at the same time. So we look at all the flowers in the progeny which bloomed at the same time, and after that we come to a final conclusion that whether it is a homozygous purple or a heterozygous purple. So moving forward, next we have the much awaited laws of inheritance. Mendel gave three laws of inheritance: law of dominance, law of segregation, and law of independent assortment. Talking about law of dominance, so as it must be very clear from the name itself, dominance means the dominant trait. It is obviously dealing with dominant trait and dominant allele. So, talking about it, each character is controlled by discrete units called factors or genes. Okay, this is so obvious that each trait is controlled by genes, which Mendel used to call factors. Okay, next, they occur in pair, and the one that is dominant masks its alternate alleles expression. They obviously occur in pair. This is visible by from here, that all the gene pairs, always the gene will occur in pair, and they have alternate forms. One is the dominant and one is recessive, and the dominant one masks the expression of the recessive one. You can see that in capital A small a pair, only the dominant color is visible and not the recessive one. Okay, so moving forward, next we have the law of segregation, which states that the two members of a gene pair or allele separate from each other during the formation of gametes in meiosis. That is, that you see, red color has a pair of genes, that is capital R, and white flower has a pair of genes small r. They would be separated. Capital R, these two capital R's would separate from each other during the formation of gametes. In and when are gametes formed? In meiosis or mitosis? In meiosis. So this is what law of segregation talks about, that the two members of a gene pair separate from each other during the formation of gametes during meiosis. Now comes the last and the final Mendel's law, that is law of independent assortment. For this, you need to have knowledge about dihybrid cross as well, which is another big topic, and I don't want to burden all of you with a lot of things at the same time. So we shall cover it in the next video. Till then, keep revising. And yes, we also provide our students free handwritten notes and quiz to check how much they have learned. You can also download our free handwritten notes from the link given in the description box, and also participate in the quiz, and you can find the link to participate in the quiz from the description box itself. For the next video, stay tuned with us, and in the upcoming videos we will be discussing about law of independent assortment and the non-Mendelian laws. Till then, stay tuned, keep learning, and keep posting your queries on our comment section, and we'll try our level best to solve all of your query. And don't forget to download our free handwritten notes and participate in the quiz competition. And yes, don't forget to like, share, and subscribe to our YouTube channel. Thank you so much.